Hello everyone, my name is Nicole Watson and I am presenting paper 37, Luminescent photoCORMs enabling disabling carbon monoxide delivery upon blue light irradiation. When you hear the words carbon monoxide, you usually think of a poisonous gas, but not a gas that can be used to help patients. Since 1999, there have been many advances in the development of how carbon monoxide is used to treat diseases. In the picture on the left side of the screen, it shows 6 different pathologies carbon monoxide can therapeutically treat and target, along with the beneficial properties. Given at a low concentration, carbon monoxide has been very beneficial. When using carbon monoxide, it is essential that we use a strict controlled environment. If you get too much carbon monoxide in the body, it replaces oxygen in red blood cells, and that leads to serious damage or death. Carbon monoxide releasing molecules, or known as CORMs, are used to get carbon monoxide in the body under a strict monitored dose control. They are bound to transition metals and act as good delivery agents to get a controlled amount of carbon monoxide into the body without affecting oxygen transport by hemoglobin. In the picture on the right, you can see the fluorescent blue ligands connected to a transition metal. The specific delivery of CORMs of carbon monoxide is delivered in the body, and the fluorescent light allows the detection and the tracking of carbon monoxide location in living cells. Other research has shown releasing carbon monoxide has helped to eradicate colorectal adenocarcinoma from HD29 human cells by using a green luminescent light to help track the distribution of carbon monoxide using manganese photoCORM. And the molecular structure of this particular manganese photoCORM is located on the right side of the screen. And you can kind of see the structure and how the molecular geometry was around the manganese metal center. It has been shown that administration of carbon monoxide in patients can help target inflammatory, vascular, sickle cell, and infectious diseases, along with malaria, diabetes, hepatitis, and it can also be used as a potential anti-tumor agent. The objective the authors were looking for in the paper, they were trying to find carbonyl compounds to release carbon monoxide closer to the visible light spectrum using luminescent photoCORMs to release the carbon monoxide under blue light. In their experiment, they used 2 new manganese photoCORMs known as complex 1 and complex 2, and they noticed that these complexes had properties of carbonyl prodrugs and released carbon monoxide under blue light. You can see on the right side of the screen the actual structures of the manganese 1 carbonyl compounds or complexes 1 and 2. The 2 new manganese photoCORM complexes each have 6 ligands attached to a metal center, giving the shape of a octahedral. Each manganese center is attached to 3 carbonyl ligands arranged in a facial coordination, 1 bromo ligand and dinitro ligands, which are bidente. The 2 new manganese complexes, complex 1 and 2, are considered to be organometallic compounds due to satisfying the 18 electron rule. The metal in complex 1 has 7 d orbitals with an overlap of s and d orbitals, but it also has a +1 oxidation state, giving the metal complex only 6 d orbitals. Complex 1 has mostly strong field ligands, so I predict it will have a low spin complex, and the electron configuration is T2 sub G6, E sub G0. The synthesis of actual complex 1 is shown on the right with the different steps showing the actual formation. Complex 1 was synthesized with oxalate H and manganese carbonyl compound to obtain a bright orange precipitate. The complex and the ligand were both purified and characterized. The precipitate was vacuum filtered and rinsed 3 times with cold methyl chloride and collected. Orange crystals were actually obtained from a slow evaporation from methyl cyanide solution at 20 degrees Celsius. Various characterization techniques were performed by the authors. I'm going to be talking about 4 of those. The IR spectroscopy measured complex 1 molecular vibrationals by recording the wavelengths of what was actually absorbed as the complex 1 molecules vibrated. The UV spectroscopy quantifies the components of complex 1. The NMR spectroscopy identified the molecular structure using peak patterns, and the single crystal X-ray diffraction identified the actual atoms of the actual crystal, their locations, and their bond lengths. The IR spectroscopies revealed facial isomers for both complex 1 and 2. Complex 1 and 2 were very similar, indicating very similar structures. You can see the arrows that I have posted, the black arrows, the green arrows, the light blue arrows. All these peaks occurred between complex 1 and complex 2 at a very similar wave number for both complexes, indicating very similar structures in both complexes. With the UV vis spectroscopy, this actually was done during the actual blue light exposure at wavelength 385. With complex 1, there was a conversion of the deoxymyoglobin to the carbon monoxide myoglobin. So at 540 nanometers, there was a peak indicated of the formation of the new species. And then there was a little bit of a drop at about 560 nanometers, indicating the carbonyl dissociation during the light exposure. And then a peak again at 575, indicating the formation of the new species during the light exposure. The proton NMR spectroscopy, again, this was done to help confirm the actual structure, but I do want to point out a few peaks. There was a multiplet at 7.75 parts per million that matched the hydrogens in the benzene ring, as well as a triplicate at 7.9, a quartet at 8.25, and a doublet at 8.38 parts per million. They all matched the carbon-hydrogen bond in the benzene ring. And then there was a triplicate at 8.49 and a doublet at 9.29 parts per million that matched the carbon-hydrogen bond in pyridine. And all these peaks, again, help to confirm the structure of complex 1. And then we're comparing it to the spectroscopy of carbon-13. All peaks are decoupled to show only the carbons. And some peaks that I would like to point out. There was a doublet at 123 parts per million that matched the benzene ring. And then there were several singlets noted at 125, 127, 129, and 132 parts per million that matched the benzene ring. This helped to confirm all the carbons in that benzene ring structure. And then there were several singlets at 137, 143, 150, 163, and 165 that all matched the carbons in the pyridine ring to confirm the structure, again, of complex 1. Now, looking at the left side of the screen, you'll notice the actual X-ray diffraction, the actual crystal structure from the orange crystal that was obtained. but the actual crystal structure itself had a distorted octahedral shape that came from the manganese carbon bonds were actually shorter than the manganese nitrogen bonds because of the strong bonding to the carbonyl ligands, and that contributed to the distorted shape. So you can see the manganese center connected to the three carbonyl ligands in a fascial coordination around the center, the metal center, and the two nitrogen ligands in the bident coordination. Oh, and I'm sorry, I also indicated the bond lengths in complex 1 of the crystal structure indicated in angstroms. During the actual blue light exposure, you can notice some peak changes that were starting to occur from the time at 0 seconds to 560 seconds, suggesting that as the peak started to widen, the carbon monoxide stretching bonds started to disappear because the carbonyl is now free in solution, and the peaks start to widen as a result. The actual mechanisms that the authors found that the carbon monoxide was released during the experiment was during the first few seconds of light exposure. You notice that octahedral shape, and then that shape changes to a trigonal bipyramidal shape that causes an elongation of a metal carbonyl bond, causing that carbonyl to come off. Once that carbonyl comes off, you notice a intermediate forming, which is the bis carbonyl, and that intermediate was confirmed with IR, UV, vis spectroscopy, as well as electrochemistry tests. But once that intermediate forms and that carbonyl comes off, the intermediate was stable for a few minutes, which allowed the authors to actually identify it. And then as more time went by, more light exposure, all the carbonyls eventually came off. And the oxidation of the metal center is indicated by the presence of paramagnetic species observed by that proton NMR spectroscopy. The results were actually pretty interesting. Both manganese complexes showed luminescence under UV light during the photoinduction process. I'm a visual person, so when I saw that significant color change referencing complex 2, I was pretty amazed. So looking at the solution before in C compared to the solution in D before and after the photoinduction, you notice a slight green color to a very strong glowing green color after the blue light exposure, which I thought was actually pretty amazing. And then the emission spectra in complex 2 shows an increase in the emission band at 508 nanometers, indicating the ligand and the carbonyl molecules are being dissociated. The authors did achieve success in finding a way to synthesize 2 new manganese complexes and use photoCORMs to release the carbon monoxide molecules very quickly by using blue light, and getting all 3 carbon monoxide molecules to be released, and also forming that bis carbonyl intermediate. During the photoinduction process, the carbonyl ligand showed this turn off, turn on properties. The authors noted that, and these properties are important because they allow for greater precision and accuracy when you're tracking the distribution of the carbon monoxide in cells. And then using confocal microscopy, it also allowed for better tracking of the carbon monoxide molecule in cells because of better imaging. And then again, upon the blue light being shown, complex 1 and complex 2 release the carbonyl ligands in addition with the ligands that they made as part of their experiment. As far as future use, the emission properties of the carbonyl compounds are important for biological applications because they exhibit traceable luminescence to the actual visible eye to show what's actually happening. The carbon monoxide molecules are being released, and that allows for greater precision and accuracy. There is a need for further research into the release of carbon monoxide molecules in order to help target diseases and be used for future medical applications and better carbon monoxide distribution in cells. Here's a list of my references that I used in my presentation. Thank you for taking the time to listen, and I hope you enjoyed it.